Hi, I'm Dr. Kurt Woeller. The title of this mechanisms video is Natural Products with Toll-like Receptor 4 Antagonist Activity. So this video is sponsored by Integrative Medicine Academy. Integrative Medicine Academy is an online academy of different courses called mastery courses. For a full list of our courses, you can go to integrativemedicineacademy.com, and you can also email us at integrativemedicineacademy@gmail.com. Here is the disclaimer for this video, understanding that the information contained and discussed is for educational purposes only. So, a recent article came out called Natural Products with Toll-like Receptor 4 Antagonist Activity. This came out in 2018. Is very interesting with regards to looking at how natural products can assist in our overall immune function. And so I want to talk a little bit about toll-like receptor 4. And before we do that, though, we need to understand what these particular types of receptors are in general. So all of us have within our innate immune system, particularly our cells like dendritic cells, monocytes, and macrophages, receptors that are called pattern recognition receptors. And these pattern recognition receptors recognize pathogen associated molecular patterns that are molecules or the molecular structure of the cell wall of different types of microbes. So, lipopolysaccharide, mannins, peptidoglycans, for example, beta glucan, are different types of molecular patterns that these microorganisms have that our immune system has recognized over time as something that we want to pay attention to, something that our immune system wants to recognize. And so there's a limited amount of pattern recognition receptors that we have. So there's a limited ability of our innate immune system to react to environmental pathogens. That's why we need adaptive immunity, which has much more adaptability and flexibility to evolve a wide variety of immune responses. However, our innate immune system is incredibly important. So, this is just an example of different types of pattern recognition receptors. Now, this particular slide comes from a book called Essential Immunology, Browett's Immunology 13th edition. Great book. I'd recommend purchasing it. And this particular example is looking at different types of pattern recognition receptors specifically geared towards fungus. So that could be candida, that could be aspergillus, for example. And you'll notice that there are different types of receptors. We have dectin 1, dectin 2, we have toll-like receptor 9, 4, toll-like receptor 2, and they're all interacting and recognizing different layers or different molecular components of fungus. So, for example, beta glucan is a very common component of the cell wall of candida, and a particular receptor called dectin 1 recognizes it. So, again, these things have evolved over time, but there's a limited amount. Now, anytime a receptor interacts with a pathogen associated molecular pattern, there is going to be a transduction signal sent to the internal aspect of the cell. So we have what are called domain reactions. These are components that occur within the cytoplasm of the cell. So the cell receptor is basically on the surface. And these transduction signals, there's different types, create downstream effects. And in this particular case, we're producing pro-inflammatory cytokines, interleukin 12, for example. So this particular transduction signal called MyD88 will have a downstream effect for the production of different pro-inflammatory cytokines. Now, another important aspect of our innate immune system is what's called antigen presentation. So dendritic cells are part of the innate immune system, and they are considered to be what are called the professional antigen presenting cells. Certain types of B cells, as well as macrophages, can also present antigen. But if we just focus on dendritic cells, they're the main cell that does most of the legwork as far as presenting an antigen. That could be a component of the cell wall of a microbe, for example, and it will present it to a naive T cell that's sitting in a regional lymph node that the dendritic cell migrates to from wherever it picked up the antigen. And so, based on the chemical signaling, we can form different types of T cell responses. So, for example, if it's a virus contaminated cell, for example, or a tumor cell, it might just directly stimulate a CD8 cell, which is called a cytolytic T cell. And that T cell will go and have a direct killing effect on that cell. In many other circumstances, we can create what are called Th helper 2 and Th helper 1 type of cells. These are CD4 cells. And these will have a broad range of effects. So, for example, Th1 will actually communicate back to the innate immune cell, triggering a macrophage to engage or increase its innate immune activity. Th2 cells go and produce or stimulate B cells to produce or transform into plasma cells to produce antibodies. So that's where we're essentially getting IgG, IgM, IgE. But it all has to do with antigen presentation, which means it also has to do with the specific antigen itself. And so one of the initiating factors then takes us back to the pattern recognition receptor recognizing a specific PAMP on a specific microbe. So this is an example of a pattern recognition receptor for Candida albicans. And as I mentioned before, there are all kinds of different types of pattern recognition receptors. We've got dectin 1, dectin 2, so dectin 1 recognizes that beta glucan. Well, toll-like receptor 4 is a major pattern recognition receptor. There's many things that it interacts with. What I want to profile though is the downstream effects, what's called the signal transduction effects that activate transcription factors. So transcription factors are proteins that move into the nucleus of the cell and then interact with the genome for the production of different types of chemical compounds, particularly production of things called chemokines and cytokines. So as you'll notice, we've got different pattern recognition receptors along the surface membrane of this cell, and they're linked to different types of what are called domains, other types of protein complexes that sit right below, and these are the initiating signal transduction domains that pick up the signal and then transfer a signal internally from there to induce transcription factor production. So toll-like receptor 4 is very much linked to something called MyD88 or TRIF or TRIF domains that then activate NF-kappa B. So NF-kappa B is a major transcription factor that induces transcription of the genome for the production of different types of cytokines. So here's a closer view of a pattern recognition receptor recognizing a pathogen associated molecular pattern. That receptor then is linked to this domain complex, MyD88, for example, or this TREF domain. It then causes a signal transduction effect downstream. It engages something called an IKK complex. That IKK complex is linked to another protein that is bound to NF-kappa B called inhibitory kappa B protein, essentially. That complex interacts with that inhibitor, essentially breaks it apart, releases the NF-kappa B from being bound up so the NF-kappa B can move into the cell and then interact with the genome. So there are many different types of receptor complexes on our cell membranes. Toll-like receptors play a big role because there's many different types of toll-like receptors. So they're found on monocytes, macrophages, dendritic cells, certain types of B cells. Okay, and there's different types of pathogen-associated molecular patterns. So for toll-like receptor 4, for example, lipopolysaccharide, for example, within the cell wall of the gram-negative bacteria, okay, is majorly linked to toll-like receptor 4. But there could be other types of pattern-associated molecular patterns, or even things called DAMPs, danger-associated molecular patterns. These are typically internal aspects of our cells. So when a cell is stressed or damaged, these molecular patterns will be released and they get recognized by different types of pattern recognition receptors, including different types of toll-like receptors. So the classic model is lipopolysaccharide. Lipopolysaccharide interacts with toll-like receptor 4. So this is toll-like receptor 4. Toll-like receptor 4 is linked to two major domains sitting below it, the TRIF and the MyD88. They both will go off in separate directions. The MyD88 interacts with something called TRAF, and then TRAF is linked to two different pathways below for the production of two different types of transcription factors, NF-kappa B probably being the most well-known or most well-studied. And then TRIF goes over and interacts and produces interferon-related factor complexes or transcription factors. So it gets confusing because there's a lot of acronyms involved, but if you just recognize that toll-like receptor 4 is a major player in this pattern recognition receptor response for signal transduction and transcription factor activation, particularly NF-kappa B. And there's a couple ways that NF-kappa B can be activated. One is through the TRIF, the other is through the MyD88. So we can see that both can actually lead to NF-kappa B production. So this particular complex sitting here below is important as well because it also plays a role in bridging the gap between the receptor and the binding domains. So this particular article went through a number of different natural products. In fact, interestingly, some of the natural products produced actually by bacteria. But I'm just going to profile a couple things here that are well known to us in the integrative functional medicine world. One is curcumin, and the other is sulforaphane, which we get from cruciferous vegetables. So curcumin is also known as turmeric, is a curry spice used in traditional Indian and Chinese medicine. We know we can use curry spice too in food for its anti-inflammatory actions. And so curcumin appears to bind what's called the MD2. MD2 are these blue dots, the MD2 proteins. So what we've got is we really have a toll-like receptor MD2 complex that then is bound to the MD88 or the TRIF. And this allows, so curcumin allows for curcumin to interfere with both the MD88 and TRIF dependent pathways. So what's happening is the curcumin essentially is binding to these MD2 proteins. So it's blocking reaction on both pathways. And so essentially through its toll-like receptor 4 effects, it's been shown that curcumin can modulate macrophage polarization. So one of the things that happens is that macrophages get polarized, meaning that they get induced into proliferation and activity. And that's a good thing. Well, we want macrophage polarization in many regards for immune activity, but too much of it can cause too much of an aggressive immune response and too much inflammation. So, for example, microglial cells are actually macrophages within the brain, and we can also have macrophages in the digestive system that could lead to colitis. So, we can get brain inflammation as well as bowel inflammation by too much macrophage polarization induced by NF-kappa B stimulation. Sulforaphane is an interesting compound naturally found in many different types of cruciferous vegetables. Sulforaphane appears to modulate inflammation by reducing production of various pro-inflammatory cytokines like tumor necrosis factor alpha. It can also reduce inducible nitric oxide that gets activated by lipopolysaccharides. So again, sulforaphane binds with the MD2, these little blue little circle proteins here. So it's blocking the linking of the toll-like receptor to the transduction domain sitting below it. Now, the biological effects of sulforaphane are similar to curcumin, but there's actually other roles of sulforaphane as well. So it moves beyond just these MD2 complexes that are linked to the bottom of the toll-like receptor. But either way, we can see where sulforaphane and curcumin can have some positive regulatory effect on toll-like receptor activity, ultimately with regards to NF-kappa B or interferon-related factor 3 transcription production. And so, in summary, the innate immune system through macrophages, monocytes, dendritic cells contain these pattern recognition receptors for particular associated uh, pathogen associated molecular patterns that are found on microbial pathogens. So, lipopolysaccharide being a classic one of gram-negative bacteria is a common PAMP that interacts with toll-like receptor 4. Okay, and so that's a normal response, but again, too much of it can be problematic. And so excessive stimulation of toll-like receptor 4 can lead to increased inflammation, leading to cellular and tissue damage. And so, as generally as part of a healthy diet, the use of curcumin as a spice, as tolerated, and the consumption of cruciferous vegetables, again as tolerated, may help to reduce inflammatory triggering through toll-like receptor 4 activation. Now, you can get curcumin, highly absorbable forms, as well as sulforaphane as a supplement, and these could have some additive effects as well, in addition to a healthy real food diet. So, I'm not making any specific claims that these are treating a specific disease, for example, but these can have modifying effects for inflammation. This is well known, well researched, and well referenced. So the information here is sponsored by Integrative Medicine Academy. As I mentioned before, Integrative Medicine Academy is an online academy that we provide different mastery courses in a wide variety of topics related to integrative and functional medicine. So for more information about Integrative Medicine Academy, go to integrativemedicineacademy.com or email us at integrativemedicineacademy@gmail.com for questions. Again, I'm Dr. Kurt Woeller for Integrative Medicine Academy. Thank you.